Okay, last thing I want to discuss about protein is their function. It's also called the activity. What do they do? And I've tried to group it into very fundamental things that proteins can do. They can bind to other molecules. They can catalyze reactions, so they can act as enzymes. And they can undergo conformational change. So even though they have native states, they sometimes have these hinges or ways of changing their structure slightly to adapt to their environment. So in response to binding, and this allows all kinds of sophisticated machines like pumps, muscle action, and sort of information processing. So I want to give you some of these examples of these three kind of capabilities. So proteins certainly bind to DNA and gene regulation. So in bluish and in greenish is the protein, shown in two different ways, either space filling or with the bonds, and then the red purple is the DNA. And the shape of the protein is just complementary to the DNA, and this particular protein is able to read the sequence of the DNA, so it only binds to some sequences and not to others. So I can bind to one gene and not the other, turning it on or turning it off. Proteins also bind to other proteins. So this is my time to talk about quaternary structure. We didn't do that yet. I don't know if you noticed, but you can have a protein chain bind to another one, and that's the native state. So that's the case in hemoglobin. That's the protein shown here. So there's two of these orangey subunits, they're called, or chains, and two of the red ones, and they form a so-called tetramer. So 4 chains together. You can also have trimers or dimers, still called quaternary structure. So they are always together, and only if they're together does the hemoglobin work. So not all proteins have that. About half of proteins have quaternary structure. And in hemoglobin, this is important because it is a mechanism so that the hemoglobin can maximize the oxygen it can transport. So it turns out these 4 chains, they arrange each other in 2 different ways. There's a tight binding and a less tight binding state. Tight binding state likes to bind to oxygen. That's helpful in the lungs, so that all 4 sites on the hemoglobin are filled with oxygen. And then as it goes to the tissue where the oxygen is needed, as the oxygens fall off, the protein switches to the loosely binding kind, and that ensures that all of the oxygens are actually delivered and not some are being brought back. That would be inefficient if it would not completely unload its oxygen in the tissues. Okay, here's another example of binding. Antibodies are proteins. Of course, they're important for vaccination. That's the whole point in vaccination, that you make antibodies for an infectious agent that you might encounter later in life. These antibodies have been described as Y-shaped. You can see that rotating molecule. It's not quite a Y, but the blue and red and yellow parts are the ones that would bind to the molecule of interest, and then the antibody can signal to the cells that there is a foreign object, foreign molecule, and then the whole immune response can be triggered. And with the vaccination, it's triggered faster than when the body still has to figure out that this is a foreign agent and has to make these antibodies, which takes a couple of days. It usually takes to get healthy after you caught a cold or something. Here's another example of binding. So this is a special protein that isn't dissolved in water, but is integrated into a lipid bilayer. It binds to sodium. It also binds to molecule called ATP. And this happens to be a pump. So it transports sodium and potassium against the way it would naturally diffuse. So it concentrates sodium on one side of the membrane and concentrates potassium on the other side of the membrane. And the energy to do that comes from hydrolyzing ATP. So that was a bit about protein binding stuff. Here's another example of protein binding to proteins. This happens when you make fibers. So in hair, you have these alpha helices, very long alpha helices, and they bind to another alpha helix, twist around it. That's called a coiled coil. So that's a very tough kind of structure. It's bendable, but hard to rip. And these form together to make intermediate and microfibrils and macrofibrils, and then you get the whole hair. As you have experienced it, hair is flexible, but on the other hand, it has some tensile strength. And that's the molecular explanation for that. And it's held together by these proteins binding to each other, or these polypeptides. There's another example of this fibrous protein called collagen. It's found in connective tissue, and again, it's a helix, but in this case you have a coil of 3 helices forming the repeating structure of the collagen. Collagen has a little interesting twist to it. One of the 20 standard amino acids, proline, is converted to hydroxyl proline, making it more soluble. Collagen is water soluble. It sort of swells up and makes these cushion type structures in between bones in joints and so on. And the fact that the proline is changed to hydroxyproline helps with that. This conversion is done with the help of vitamin C. And if you don't consume vitamin C for a long period of time, like sailors did when they crossed the ocean and didn't know about vitamin C, you get a disease called scurvy. And you can reverse that by giving a vitamin C-rich diet. The way the sailors solved it is to have lots of citrus fruits on board when they went for the long trips. Okay, here's a little tidbit about disulfides and hairstyling. So hair gets stabilized by disulfide bridges, keeps the hair in a certain shape. If you open up those disulfides, then you can shape the hair, and then you can put them back, zip them back together. And so you can either straighten hair like this, or you can curl hair like this, and the straight hair or the curly hair will stay like this for a while. So in hairstyling, you have a reducing agent called ammonium thioglycolate that breaks the disulfide bridges, and then you have hydrogen peroxide that reforms them again. And those are 2 chemical agents used in hairstyling. Okay, so that was a lot about binding. And the next thing is that we can have proteins that catalyze a reaction. That is a separate topic, sort of its own chapter, and so I will stop here, and the next time we'll talk about enzymes and how they are able to catalyze reactions that would otherwise not happen at all, or just happen very slowly. Thanks.